Hi, my name is Jared Lettery, and welcome to the AI Academy. Our next speaker is Dr. Mike Large. Mike's currently at Cooper Surgical. He has his PhD from Baylor University and is deeply involved in the PGT program at Cooper. But our topic today that he has considerable experience in developing products will be identification using a variety of tools to maintain continuity in terms of identification and also parental backgrounds. Mike, thanks so much for participating. Look forward to your comments.
Hello, everybody. I am Mike Large, and I work with Cooper Surgical. And today I'm going to be talking about we've done there with RFID technology and genetic assessments and the application of AI to those technologies and how we're leveraging that to assure sample identity and assurance through the lab, And so I'll be looking through a few topics today and really start off with a brief introduction to RFID technology or radio frequency. And then I'll be adding some insightful information on how we're leveraging intelligence and AI and how we're applying that to that data and really seeking to add value and benefit there to improve operational workflows. Then I'll pivot towards an introduction in artificial intelligence in PGTA, or pre-implantation genetic testing for aneuploidy, and how we've been able to build on top of that technology and really assure sample identity and family assessments through genetic components. And then finally, I'll leave a few discussion topics for you to mull over as you go back and really discuss some of your thoughts on this with your colleagues. And so here I have a really nice image of a basic IVF laboratory, a modest sized one at that, and I'm going to walk through some of the complexities here. And so if you're familiar with the field, that should be really no surprise, but as you stack it all up, it actually does become quite surprising. And so start off looking at the workstations, and you can see between the sperm prep, ICSI, incubator storage, and embryology that there are, in fact, many workstations involved here. And of course, in any given lab, there are multiple embryologists and scientists busily working throughout the environment. There are likely multiple dishes. You can see them there in the ICSI prep and the incubator and the embryology hoods. Multiple tubes, perhaps for the sperm and the ICSI prep, some of the embryology and biopsy tubes as well. And then there are often multiple IVF patients and specimens in the lab at any given time. And so obviously this presents a significant operational challenge to ensure that everything's being done appropriately and that no mix-ups occur. Historically, this has been done predominantly through human witnessing, where some embryologists will look over the shoulder of others to try to ensure that everything is being done accurately and the identities are matched and checked. But as complexity and all of the things going on in the lab continue to escalate, IVF labs grow and become even busier, this becomes a challenge. And we certainly saw through COVID and social distancing that there was also an increased difficulty with human-to-human witnessing. And that's where electronic witnessing comes in. And so here in that same lab, if you're able to add electronic witnessing, and I'll walk through some of how this works in the various different workflows and stations, you can actually leverage technology and equipment over human checking of errors and mix-ups. So here's a simple workstation, and ultimately what you should note is that RFID readers are built into the equipment, and so you can electronic fields that are capable of measuring small tags that are placed on all of the plastics and consumables that once used can be associated with a given patient or a given sample. And so in a very seamless and sort of automated way, we can have this dish come in here. Let's turn on my laser for a second. We can have this dish come in here, and as we move it, excuse me. As we move it into the workstation, you can see that on the screen, it automatically identifies who the patient is and what the samples are there. Now, if you were working with sperm files from a different patient, and this happened to be the wrong patient that has entered the workstation, you get automated visual and audio cues that something has gone wrong. This is also true if you've built in workflows of standardized procedures in your labs and you know that certain steps are supposed to happen in certain orders. And if you've done something in the wrong order, the same type of phenomena will occur. So really all sort of built in and happening behind the scenes. And the way that this is ultimately delivered is you have the work area. It's sort of the active brain and sort of communication element involved is that small tablet that's always communicating with the devices and the users. We have a managing component where you can set up some of the workflows, and I'll walk through some of the specifics here, but can set up your configurations and workflows and as well as print some labels for human readable labels. And then all this information needs to be stored. Historically, this has been in servers, so with sort of on-site databases that help capture all of this data. So when we look at the various elements of electronic witnessing, what we do see is that, of course, in that work area, I went through the principal event being mismatch detection. That's the main reason why you might leverage this technology. But you also are able to capture timestamps when things happen. And so this is really foreshadowing, and where we'll go with this is really understanding how long things are happening, taking to happen, and what it looks like there. Certainly, you can capture images and other elements as well here. And then on the manager front, as I mentioned, you can look at configuring your protocols and various different rules and preferences, some of those human readable printing and labeling, some reports and patient management, etc. And then all this is, again, stored in the database where the repository of information is, and it can be connected to various EMRs. And so once we began to really work with this and the data was coming in, we started to recognize that there's some value in this data. And if we're able to capture, for example, how long embryos are outside of the incubator, might we be able to correlate that to outcomes or to blast rates, et cetera? And so really what we sought to off of these limited servers and move it into the cloud where processing could occur. And of course, very, very simply put, you can use this to monitor utilization and capacity. You can look at standardization, as we're seeing more and more IVF practices will have multiple labs, and trying to really get harmonization on those protocols and procedures is really important, especially if lab directors or lab management are off-site. And so in this context, the information is being really uploaded to that cloud. It allows that sort of monitoring that can be very helpful for audits and inspections, as well as part of any sort of quality management systems. And then, as I mentioned, really trying to understand which procedures and what the critical timing steps are is really helpful in trying to improve outcomes over time. And so what we've really begun to work on is converting this information back into value and reporting it back into dashboards that can be customized and used to really understand what's going on in the lab, how things are going. You can track things month over month, time over time, to really understand how things are performing. And while it's really early days of putting these tools in place, we do think that there will be a significant potential to learn about critical steps and best practices in the IVF lab. And so from there, I'm going to switch to introduce some of the genomics technology we've been using. And so I would say PGTA is really a technology or an approach intended to screen for the appropriate copy number of chromosomes. So we all inherit 2 copies of every chromosome from mom and dad. We should have 22 somatic chromosomes in 2 copies, and then the sex chromosomes X and Y. And what we know is that the improper inheritance of that chromosome number, known as aneuploidy, is associated with adverse outcomes, miscarriage, and live birth syndromes. And so the principle goes that if you're able to select the embryos that have the appropriate number of chromosomes, you're able to accelerate the time to a successful pregnancy and minimize losses. That technology is really, since around 2014, moved towards next generation sequencing. And when that technology first came out, we would sequence small fragments of DNA. We would determine what their base composition is, and then we would scan the human genome and say, where did this piece of DNA come from? And you're able to tally those counts and determine on a count by count representation how evenly dispersed your data sample is. And so that would then be converted into these plots. And so you can see that at 2.0 copies, you have all of the little green dots, and the green dots represent 1 megabase bins of our 3 billion base pair genome. And you can see in this example, going all the way across each of the chromosomes in this column colors, a nice representation of 2 copies, and we get to chromosome X. There's only 1 copy in Y as well. So this is a euploid male. Pretty easy and straightforward. A leap compared to previous technologies because of the 1 million data points and the sensitivity involved here. However, not all data is as easy to work with as this. And so what we do find is that biopsy quality and the DNA within those biopsies can be challenging to read. And so independent of how much expertise and the validations that have been done by lab, reading data like this can be quite a challenge. And so you might see that degradation at the telomeres, that you're seeing here. And really you need to decipher, is that a true deletion or is that noise within your sample? And in the graph below, you can see potentially that there are intermediate copy numbers, could be mosaic chromosomes, or when you're looking at the entire genome profile, this could be variability in noise in the assay. And so after a few years of running this, we really looked to ourselves and we said, we have a very sophisticated technology looking very closely at the DNA. And we have a million data points, and we take this data and we convert it into pictures to argue over subjectively. Like there must be something better, we must be able to do better. And so that's really when we decided to build out our own platform. And one of the challenges we have in subjective interpretation is trying to decipher which signal is real and which signal is noise. And so rather than beginning on validating something as simple as cell lines, which present their DNA very well to analytical platforms, we worked our way backwards. So we, over time, we collected outcome data, and we had over 1,000 biopsies with sequencing data that ultimately we knew led to live birth or late ongoing pregnancies with no indication of any sort of genetic abnormalities. We then, as I might speak to, really developed machine learning based algorithms to sort of train our data against. And so ultimately what this did is we were able to establish the normal distribution of signals associated with those healthy live birth outcomes. And then we can test a sample data on top of that and really determine if it's differentiated from itself. We're also able to deploy some convolutional neural networks, which were really helpful in our sex chromosome classification, because unlike the somatic chromosomes, where 2 copies is always good and 1 copy or 3 copies are bad, with the sex chromosomes, you can have 0, 1, or 2 copies, all representing the appropriate or inappropriate number of chromosomes. And so the CNNs really accelerated in that setting. We also trained the platform with PGT-SR patients. And so these are patients who have small segments of chromosome that have broken off 2 different chromosomes and reattached back on the wrong chromosome. And we know that through karyotyping, we can identify patients who have these conditions. And it's a 50-50 chance if the embryo has inherited the imbalanced form of those structures and duplications with verified breakpoints that we can use to train the system as well. Not only what we sought to do was deliver, rather than subjective interpretation of images, a statistical robust platform that made automated calls that could be reproduced time and time again, and removing that subjectivity and avoiding the possibility for human interpretation and transcription errors. And what we're really happy to see is that over time, really within the first year, we went back and looked at our change in ploidy classification rates, and across all age groups, we saw an increase in the net euploid classifications, meaning that patients had more embryos available for transfer. And this actually meant that about 5% of patients who didn't have a euploid in the past would have had a euploid available for transfer. So pretty significant impact. And this is, of course, with the trade-off of calling fewer mosaics. And the aneuploids ultimately are, when we see a reduction of aneuploids here, it's because we call complex mosaics, where there are multiple mosaic chromosomes as an aneuploid. And so as those change, they also into euploid. And knowing that AI and machine learning really thrive from big data, we really didn't want to rest on our laurels. And so that sequencing platform was built out on the MiSeq, the same as those subjective interpretations. And you can see that the flow cell here is where the sequencing occurs on. And really, there's a small glass groove within that flow cell where all the sequencing occurs. By comparison, here's the Illumina NextSeq, and you can see that there's significantly more real estate to do a lot more data on. Further, you can read a sentence in one direction, and we'll call that single-end sequencing. And certainly that has its benefits, especially in terms of time and accessibility. But the human genome is full of repetitive DNA that can be difficult to map. And so as a metaphor, you might think of trying to find the phrase "winter is coming" if there are any Game of Thrones fans out there. It would be very redundant throughout that whole series. It's something that's said time and time again. And if that's the sentence that you read, you would have no idea where in that television series that sentence came from. However, if you read a couple sentences later in reverse, you would find actual unique information that would then allow you to map that DNA. And so we call that paired end sequencing. And so we also increase not only our depth, but also our pairing to give us a net 8x increase in the number of raw data going into the system. This also at that point allowed us to begin what's known as SNP calling. And so SNPs are just a single base pair change. We have them, they're very useful to us geneticists in that they occur approximately 1 to 200 base pairs. And so we have a lot of them. Many of them are not associated with any known phenotypes or conditions, but some of them are. For example, it's a SNP that causes blue eyes. There's also a SNP that phenotypes, but the vast majority do not. However, it's sort of illustrated here in the schematic that can be very useful for biological markers. So we typically classify SNPs as the major and minor isoforms, the ones that are most common in a population and those that are least common, A being the representation for the major isoform. And we can have different SNPs inherited from mom and dad, and there we call that heterozygous, one of each form, or we can inherit the same SNP from mom and dad, and they're homozygous major, AA, or minor, BB. And so why this is useful is when we look at the heterozygous SNPs, the ratio of A to B should be 1 to 1, or 50% B, as an example. And when we look in the cells that are illustrated here, we look in the monosome example, we can't, when we look for heterozygous alleles, there are none. We have a complete loss of heterozygosity. And so the SNP ratio is telling us that there's a lack of information. And in the trisomy example, if you look at the second SNP there, where we see reading from top to bottom BAA, instead of that 1 to 1, 50% ratio at the heterozygous locus, we actually have 2 A's to 1 B. And this drift in ratio informs us that there's a gain of material. And I'll show some data later, but as I've color coded here, if you're looking at what SNPs are or are not present, this can also help you confirm some identity. And so for the first time, we're really able to combine our copy number assessment using machine learning in an automated calling platform with a secondary level of information with the SNP assessment and SNP ratios. And showing again some data here, we have a loss of chromosome 18. If I plot out the data from our AI system, you can see it there. And when we look at the parental information, and so in this graph, the way you interpret it is each dot is a chromosome, and shifting towards the left, that chromosome appears to be more like dad, and shifting to the right, it appears to be more like mom. And so in this case, we see that there's a loss of chromosome 18 in the copy counting, but we see that the chromosome that is present looks like mom, so that the monosome is in fact paternal origin. We're missing dad's copy, and that's why the one copy remaining. looks like mom. It's also beneficial that when we look at from a purely quantitative standpoint, and I talked about that bin normalization, do I have even read number at every single bin across the genome? And in this case here, we see, okay, I have even read number, even and sex chromosome X with nothing in Y. I think that this is a euploid female by classic NGS techniques. However, when I add SNP ratios and parental information, I can see that in fact, every single sample is skewing towards maternal. And what this ends up telling us is that we have maternal triploidy present. And this is important. It occurs approximately 1% of blastocysts, and then it can lead to molar pregnancies and miscarriage and adverse outcomes as well. And so being able to detect it. And we saw in a retrospective observational study shortly after launching this platform that in fact leveraging this approach increased ongoing pregnancy and live birth rates. And of course, that was at the expense of biochemical losses. And so not only were we beginning to call more euploids by increasing specificity, but by repressing those noise levels, by adding female triploid detection, we were able to actually decrease losses as well. We still didn't rest on our laurels, and I talked about starting with the MiSeq and the NextSeq, and recently we moved to the NovaSeq, and really the goal here is we know that even in our better prognosis patients in good IVF centers across the world, success rates plateau around 70-75%. And so really interested in why do the remaining 25 or 30% of euploid blastocysts fail. And one potential reason is that we know that PGTA today is a 5 megabase, 5 million base pair limit. And so we're interested in looking at a finer level and trying to correlate any changes in either the base pair composition or smaller regions to pregnancy outcomes. And moving to this platform, we're now over 22 times the amount of data as the original NGS. And again, we can try to look for things. We can take our sequencing data and we can plug in health outcomes and look for patterns and tell it what to look for. We can do unsupervised learning, and we can just track the data and find out what comes out and if there are any associations that we might not have predicted in the past. And so that's really what we're looking forward to. And with the last couple slides, talk about that at the same time, we were interested in investigating non-invasive PGT. And so recently it's been discovered that IVF culture media contains DNA from the embryos, and maybe this is a suitable specimen to look at. And so here on the left, you see a graphic representation of our data for a biopsy. And you can see it's a female euploid. And when we look at the media, again, you can see, okay, this profile, this DNA is of pretty similar quality. And we're seeing female euploid here. However, when we added our parental assessment and our SNP calling, we look in the biopsy, we see nice biparental inheritance and equal representation. But when we look in the media, we in fact see everything is skewed towards mom. Not shown here are the SNP ratios, but when we look at SNPs in this profile, they are in fact 1-to-1, 50% heterozygous. And so it's not actually a female triploidy, but what we infer here is that it's a maternal cell contamination, potentially from accumulus follicular cells. So that was an ultimate reason why we think that non-invasive approaches aren't quite ready for clinical use. But while we were doing this research over the last couple of years, at the same time, we began to see IVF mix-ups. And so you don't need to sort of belabor the point and read through each one of these titles, but it's obviously been a real tragedy, and it's been very difficult for the field of patients involved to really go through this. I've already talked about electronic witnessing in the lab, and I'll talk about what we built out from the genomics perspective. So ultimately, as we saw in that non-invasive work, using parental DNA, it really was powerful for us to be able to understand the validity of that biopsy. And so what we sought to do is on the top, I have what I'll call a true trio. So know that I have mom and dad samples, and I compare them to an embryo, and they're the right information. Because our platform is cloud-based, we can actually assign a different parent to the embryos, as an example. So in the second example, I assigned the wrong mom, and in the third example, it's the wrong paternal contribution, and in the final row, we have both parents were wrong in the blue color there. And we can see what our data looked like and how that performed. When we look at our validation data, did figure here, but really it's simple if you break it down into its components. And so on the y-axis is that maternal versus paternal balance. We've discussed that with the pattern of origin. And so shown in pink or red, there those samples are looking a lot more like mom, which means that you probably have the wrong dad, vice versa in the blue. And in the green, we have that equal balance. However, if you have both parents being wrong, then the embryos are not likely to look more like mom or more like dad. They're equally unlike them. And so we needed to incorporate novelty. So if the embryos have SNPs that are not present in mom or dad, we see an increase in novelty scores. And while this is just a graphic representation of what the algorithms do, you can see that we have nice separation of those data sets. And ultimately, through nearly 3,000 permutations, we have extremely high accuracy, sensitivity, and specificity. Certainly can help on the genetic component to link and assure that these samples are related using technology. And so finally, I think it begs the question of, should proactive electronic witnessing and or genomic mismatch detection be considered the standard of care? And what are some of the best practices? And to what extent does that mitigate liability if you're able to prevent errors before they occur or check them in live time? And also, what are some of the key data elements when I was talking about using the witness QC and feeding into it? I talked about time as one of the obvious ones, but maybe temperature variations, of course, could be another key variable. And ultimately, the AI is only as good as the validity and the type of data that go into it. So what are the key elements of data that we should be capturing as part of our typical workflows and streams? And then finally, when we look at the benefits for providers and clinical practices in the IVF lab, health outcomes, what are some of the hurdles in doing this? I think that some of the value hopefully begins to speak for itself. But I think there are also challenges associated with, do our electronic systems talk to each other? Are there APIs and how do I know it's accurate? Am I relying on humans to plug the information in, and is that error prone, et cetera? What are some of the challenges that we have to overcome for these approaches to really be leveraged from a higher order of AI and intelligence around it? And so with that, thank you for attention. And if you have any follow-ups or would like to contact me, my email is included here. And thanks again.
Mike, this is Jared Lettery. Mike went far beyond just talking about witnessing and extended the discussion into the PGT, but did circle back and relate how PGT can also verify parentage. We won't talk about PGT. I'll make a few simple comments. The PGT debate could fill a Dickens novel, but I did want to comment about a couple of things. The witnessing program that he describes is transitioning the lab as it's currently practiced into the lab of the future, transitioning from qualitative assessments, double witnessing, people commenting, documenting handwritten notes that, in fact, biopsy from embryo A went into prep A, et cetera, to something that's quantitative and documented and expands a database. And anytime we can transition from something that's qualitative to something that's quantitative, we then have the ability to measure and track. And with that ability, we can improve outcomes. The other issue that I'll mention, and then we can end, is that I think up ahead, as over-the-counter, direct-to-consumer genetic tools expand, everyone's using them. Uptake will be tremendous in the future. I think we're going to have people searching for parentage if they were conceived in an IVF lab. This is somewhat speculative on my part, but we're already starting to see some cases. I think the initial one was back in 2019. There were previous cases, very isolated in the past, but we're going to see more and more cases where genealogists are tracking the origin of anyone conceived through ART to make sure, in fact, that the parents are their parents and that there was no confusion in the lab. So to have a quantitative and electronic witnessing system, I think, will be an essential part of the lab in the future. We're not even talking about the time saving and the manpower saving that these tools can afford a lab. As I mentioned in the earlier comments at the start of this may not add to the bottom line. It may not necessarily improve outcomes, but I think it's going to be part of the tools we need to run a lab efficiently and quantitative in the lab of the future. Thanks so much.